we'll go ahead and get started. So for our Data Wednesday seminar today, I'm pleased to welcome Dr. Matthew Hahn from Indiana University to talk about the evolution of mammalian mutation rates. He earned his BS degree from Cornell and got his Ph.D. from Duke under the mentorship of Mark Rausher, and he was an NSF postdoctoral fellow at the University of California, Davis, with Charles Langley and John Gillespie. He is a distinguished professor of biology and computer science at Indiana University, where he has held a faculty position since 2005, and he currently directs IU's Center for Genomics and Bioinformatics. His research focuses on how evolution has shaped organismal and genomic diversity and how adaptation is achieved using multiple different types of molecular changes. And his lab couples empirical studies of genome sequences with the development of mathematical theory, new statistical models, and the implementation of open source software. And with that, I will leave it to Matt to take it away.
Awesome. Thank you, Kelsey. Thank you, everyone. I'm going to give a new talk today, one that I haven't given, but that summarizes a bunch of work that we've been doing. And so I thought this would be a nice chance to try it out on you. And because Zoom talks suck so much, you should please interrupt me. Otherwise, I will just be talking to little black squares for the next 50 minutes or so. Okay, so what I am generally interested in is the genetic basis for evolutionary change, right? That is, if we have current species, how did each of them change since their common ancestor? There's a reconstructed chimp-human fossil there. What were the genetic changes that occurred on each lineage? What were the evolutionary forces that affected those genetic changes? What types of mutations were they, etc. The particular trait I want to talk about evolving today is the mutation rate. Okay, so if we imagine that Darwin had a kid and that kid had some number of mutations, that number of mutations is a trait that evolves on a tree. It evolves on phylogenetic evolutionary trees. And so we can see this kind of at a very broad scale. If we think about the variation in mutation rates among the domains of life, they vary. All multicellular metazoan things have a relatively high rate. Unicellular eukaryotes have an intermediate rate, and then unicellular prokaryotic things, eubacteria and archaea, have a very low rate. But there is variation, and there's a lot of variation even within smaller timescales as well. So mutation rate is a trait that varies among species, and of course it's also an important trait. So here's just some reasons why it's important. It's important for disease. The gene CFTR, mutations in it cause cystic fibrosis. There's over 1,000 known mutations in this gene that cause different forms of cystic fibrosis. So understanding mutation is important for disease. Understanding mutation is important to understand why sex is maintained. So lots of theories about sex, and in particular ones that invoke Muller's ratchet, basically are invoking mutation as a cause for the advantage of sexuality over asexuality. Understanding mutation is important for understanding adaptation. So there's a distinction between adaptation from new mutation that just happens once, so that's a hard sweep, and possibly new mutations that happen multiple times, so those are soft sweeps. So if we want to know, well, if there's a new environment that we encounter, how does adaptation happen? We'd like to know something about the mutation rate. How likely is it that we'd get multiple mutations both to the same state that would be advantageous in this new environment? And of course, mutation is important for molecular evolution. So this is a famous pattern that I'm showing here, the hominoid slowdown, which is that the nucleotide substitution rate is actually slower in the hominoids, humans and chimps, than it is in the old world monkeys, here baboon and macaque, and that's slower than, again, the new world monkeys. This is a bit of an exaggerated tree, but you get the general idea that there's this relationship between the different taxonomic groups and the substitution rate, and we have some idea that that should be tied to the actual nucleotide mutation rate if it changes so much among species. So mutation rates vary and they're important. The other reason I think they're interesting is that they're quite complex, right? Mutation rate, even though we represent this trait as this one letter, mu, it's actually made up of all of these little traits, right? Mutations are caused because starting from the zygote, there are cells that are at first not a germline, and they're dividing and mutating. they're eventually set aside as germline stem cells, and then they go through several more rounds of replication before they finally undergo either spermatogenesis or oogenesis to become a gamete, right? So when we say mutation rate, really what we mean is the rate of error at all of these different stages of development. And so it's kind of like saying, the trait of height is really the trait of, heel bone height and ankle bone height and shin bone height and all of those, except it's a little bit less allometrically constrained when it comes to mutation. So it's a complex trait, it varies, it's important. And I want to know, how does it evolve? Right? What are the different forces that make the mutation rate evolve? How fast does it evolve? And so the obvious thing to do, if one wanted to answer this, would be to go out with your sequencing machine and sample a bunch of species. And you wouldn't just sample one individual, you'd have to consistently sample a trio. So in order to get the mutation rate, the most straightforward way to estimate the mutation rate is to sample 2 parents and an offspring. And what you're essentially looking for, I'll talk a little bit more about the methods, is alleles that are absent from both parent and present in heterozygous form in the offspring, because that means somewhere along the way there was an error that was inherited by the offspring. So that seems like an easy task. We have lots of sequencing power. It's relatively easy to get a single trio of most organisms out in the wild. The problem becomes, when we have those data, how do we start to compare different species? Right? So we have now a per generation mutation rate, the number of mutations that were inherited by an offspring per base, controlling for the genome size, for all of these different species. But per generation mutation rates vary with the lifespan of the organism. So here's a little graph that we put together, kind of showing just among multicellular organisms, how many mutations do they leave to their children and how long are they alive? And I think obviously there is a relationship, because the longer the parents are alive, or at least alive before they have kids, the more mutations can accumulate in the germline, right? Or if they don't have a sequestered germline like plants, just the more mutations can accumulate. So if I start comparing species with different generation times, I'm going to obviously see a difference in the per generation mutation rate, simply because I'm comparing very different time intervals. And so you can kind of imagine that if I'm comparing a Drosophila mutation rate and a human mutation rate, there's probably a lot of differences in how long development takes, how many cell divisions there are in development before you set aside the germline stem cells, and all of these different steps along the way, how long gametogenesis occurs for. Drosophila are having kids at 2 weeks of age as opposed to 30 years of age, right? So that's a huge difference, and we have to understand, okay, well, if I want to know how fast mutation rates evolve, I have to understand, how do I compare them? An analogy I will try to invoke is tree height, right? If we compared 2 tree species and found that these 2 individuals were different heights, we might say, aha, this is like the characteristic of our species. They're just different. But if I then told you, well, these trees happen to be of different ages, you'd say, well, maybe if I was able to measure them at the same time, they'd really be the same height. And in general, we call these function value traits. So our trait is a function of age, right? It doesn't seem fair, if we have a function value trait, that we would compare them at some different value of this very important variable. I have a coffee, so I'm going to take sips of coffee too. And so we know, in fact, that mutation rate is also a function value trait and is a function of age. So this is a graph from a bunch of Icelandic trios, where each point is an offspring and the number of mutations that offspring inherited. And along the x-axis is the age of the father at conception. So the mother has a different age, but they're correlated at least. And what you can see is that if I just measure the mutation rate for an older father, it would be somewhere up here, right? A father of 40 years of age, they'd have 80 mutations that they passed on to the kid. And if I measured a father who had a kid at 20, there'd be many fewer mutations, right? And we don't want to say that the mutation rate evolved between these two individuals. Nothing genetic necessarily happened. We're just measuring them, this function valued trait, at very different points in time. So in humans, not only is mutation kind of function valued overall, but it's slightly different. The function that determines mutations is slightly different between males and females. It does go up with time in females, but it goes up even faster for males. So males leave about 75% of all mutations, and that slope of that line that goes up with male age is steeper for males than females. The reason, so there's multiple hypotheses for that, but the reason that I'll stick with and our data seem to also support is that both males and females have a relatively fixed number of cell divisions during development before the germline is set aside. You can see the numbers at the top. Maybe they differ a little bit. Males have 30, females have 22. Those numbers themselves are guesses from the size of the actual testes and ovaries. But after that point, female mitosis stops. And when males hit puberty, it starts again. So a male at 40 has gone through many, many more rounds of mitosis and therefore of genomic replication than a male at 20. So that's why we think that rate is going up with time, especially rapidly in males. Okay, so we have to imagine that we want to go learn about the mutation rate, and what that means is that we need a way to compare mutation rates among species, right? I'm going to get, you'll see, I'm going to get data for a mutation rate from a monkey that had kids at 6, and I want to be able to compare that mutation rate to a human who had kids at 30, and I want to be able to say, did something evolve about the mutation rate, other than the fact that I've just compared two organisms at very different ages. Okay, so I'm going to try and answer that question, and first I'm just going to back up and say, we want to answer lots of questions about the mutation rate. And so first we want to know, what's the function that allows us to compare species? Once we have that out of the way, once we can compare species on some ground that makes for fair comparisons, then we can ask, has the mutation rate evolved between species? If it has evolved, what stage or mechanism has changed? Is it increased number of cell divisions? Is it the proteins that are involved in proofreading? Is it something else, the environments they live in? Are different stages or different mechanisms more labile than others? If we can look at evolution along a bunch of different lineages, which of those change, et cetera. So lots of questions about the mutation rate that we can ask. And they all start with asking, with figuring out this function that allows us to compare species. Okay, so here's the outline of the talk I'm going to give. And I'm going to talk about data sets from 4 species and tell you about how we collected mutation rate data from the first 3, how we're going to use that information to say something hopefully intelligent about humans at the end. But I'll start with owl monkeys. So how do we compare mutation rate? That's the first question we have to be able to answer. So we have monkeys of 6 years old, humans of 30 years old. And now it won't let me change slides for some strange reason. Let's try this again. Okay, so what's the function that determines mutation rate? So it seems obvious that this is the function, right? We can learn it from humans if we just assume it hasn't really changed between humans and the other primates, then this is it. The problem, of course, is that this function only goes down to 15 years of age. It doesn't go above about 45 or 50. It doesn't go below 15. We're going to ignore the upper half because there's not too many primates that have kids after 50. So really the question is, what does the function look like when it's lower than this bottom level of the human rate, right? We can only estimate, if we're using trios, we can only estimate the per generation mutation rate from humans at an age at which they have kids. So we're limited to about 13 to 15. So there's 2, I would say, main competitor models for what this function looks like. One I'll call the total longevity model, and that just says, hey, it's like a big straight line, right? There's some overall mutation clock that's going on. Mutations are just ticking away at some rate, and that if I went out and measured the mutation rate in a 6-year-old monkey, that it would be right on this line. Just keep drawing it down towards the origin, right? So this model says it's a time since conception. That's the main determinant, right? In order to adjust among species, I just say, well, how old are you, since you were conceived? The alternative model considers the fact that there's these two very different stages of germline replication. One, which is pre-puberty, right? The set number of cell divisions, at least within one species. And then once they hit puberty, there's this new increasing number, right? So in the reproductive longevity model, we imagine, yes, the kind of rate of increase is exactly the same in these other primates as in humans after puberty, but they hit puberty at a much younger age, right? So let's just assume that among primates, it's probably, gestation times differ, but maybe the number of total cell divisions to get to develop the fully developed organism is about the same. So maybe the number of mutations at puberty is about the same, and then the clock starts from there. So under the reproductive longevity model, it's really the time since puberty that's the main determinant of the per generation mutation rate, plus the mutations that accrued before that point. Okay, so these are the two models we're going to contrast, and to do that, we're going to study owl monkeys. So we actually started this by basically asking our primatologist colleague, Jeff Rogers, do you know of primates with a sequenced genome and pedigrees where we are pretty sure of paternal-maternal relationships, and we know the ages at which everyone had their kids? And so owl monkeys, this colony down in the Bastrop Primate Center in Bastrop, Texas, happened to be the first hit that had lots of available individuals. So we had about 30 total individuals from two different types of pedigrees. The pedigree on the left, 3 generations with 1 F2 grandchild. And then the pedigree on the right, there's 2 F2, 2 grandchildren. And I'll explain why those F2s are useful as well. Okay, so some technical details about the genome sequencing and the finding these needles in a haystack. I should say also that all of this was led by my former grad student, Greg Thomas, who is now a postdoc with Jeff Good at the University of Montana. So you can ask me more details if I don't cover the ones you're interested in. But essentially, we sequenced 30 individuals to 37x coverage on average. So using Illumina, the important things for finding mutations are making sure that the mutation you're calling in the kid is real and not a sequencing error, and making sure that you have enough coverage in the parents that you're not just missing a heterozygous site there, right? So 37x coverage helps to do that. We still had to filter based on lots of things. Allelic balance, so if they're truly heterozygous in the kids, it'll be about 50-50. Somatic mutations won't be at 50-50 unless they're happening in the very first few cell divisions of development. Filter on read depth again, so that we can be sure about, and genotype quality, so we can be sure about the absence of mutations in the parents and the presence of real mutations in the kids. And then the F2 individuals are especially useful because if we're calling mutations correctly, half of them should be inherited by the offspring, by the F2 individuals. So half of the mutations we call in the first should be called. So that's always our nice internal check. Are we doing this correctly? Yes, because we see basically exactly half of our mutations being passed on. Okay, so here's the mutation rate. We do a lot of work. We sequence many, many billions of bases. We find a few hundred mutations among all of these trios. And the mutation rate is 0.82 times 10 to the negative 8th per base per generation. So that's really good. That's less than 1 error per 100 million bases. Like the proofreading machinery is really good. And it's still, even though we've gone through so many replications to get to a fully grown individual. And indeed, just as in humans, males produce 70% of all mutations. So I didn't go back and show you, but we can also use the F2 individuals to phase mutations because we can see when they're inherited by an F2, which parent they came from. We can assign them as either the maternal or paternal parent. Okay, so what is the function that determines mutation rate? How do we use the owl monkey data to help us answer this question? So these are, again, the data from human Icelanders. We have about 43,000 mutations here. And we can parameterize the model, right? We can basically fill in this orange line. So that's what the human line looks like. And we can ask, well, where do the owl monkey data fall? Do they follow the total longevity model or the reproductive longevity model? And it turns out they follow the reproductive longevity model quite well. So again, you can see that owl monkeys leave more mutations with their offspring as they get older, and that the linear fit through the owl monkey data is a pretty good match to the prediction from the Icelandic individuals, right? So I just basically took the Icelandic line, shifted it so that its puberty started at 1 instead of 13, and that's the fit. If we were to look at the total longevity line, it would be much further down below, right? This is much too high for that. So the owl monkey kind of mutation rate, the owl monkey mutation rate is being set by when puberty starts, not how absolutely old they are in years. Sarah stopped walking, so now I'm like, does she have a question?
I do. So all you're saying is the slope of the line is the same.
Not just the slope. So the slope would be the same. Actually, the slope doesn't have to be the same. So that's one amazing thing. Actually, let me just point out, like owl monkeys are accumulating mutations at basically the same rate as humans per age. But they're also starting at a point at puberty, basically at the youngest owl monkey, 1-year-old owl monkey has as many mutations as a 13-year-old human. And then, the mutations start accruing more after that.
Okay, maybe I'm misunderstanding, because when you set up the two predictions graphically in the beginning, what I was just thinking is that one of them, they have the same slope and intercept, and the other one, they just have the same slope, but a different intercept.
Yes.
Because they have a different age at puberty. So now I'm trying to distinguish, do your data support the same slope but different intercept because of the difference of the age of maturity in owl monkeys?
Yes.
Or is it a third case altogether?
No, it's that case.
Okay.
All right. So
and what about?
Yeah, go ahead.
And I know that this will be a function of the number of data points you have, but does it look like the variance around, is it similar?
It's really hard. I mean, we have so many data.
You've got 42,000. Yeah. Okay.
It's always messier for our data than anything you can collect in humans.
I'm just, so interested in intraspecific variation. So I'm curious how tight this.
We cannot say anything smart about genetic variation within species.
Okay.
Yeah.
But you're saying the puberty intercept is essentially the same.
Yep. The puberty intercept is exactly the same. Yeah. It does not keep going down.
So Matt, just to clarify, when you took that line from humans, you slid it horizontally across the graph. You didn't slide it down the slope.
Yep. Exactly right. Yeah, that's a good way of describing it. Yep. Okay, so that's what it matches. It's this purple line over here, not the extension of the orange line. I'm going to skip this because it's too hard to explain. Okay, so in general, for this one species, we support the reproductive longevity model, right? So owl monkeys are little humans that just started puberty at a different time. So for this one species, it seems like reproductive longevity is the right model to understand how mutation rates are changing. So just conclusions for this little part, there are more mutations as owl monkeys age, especially in males. This rate of accumulation appears to be the same or not significantly different from the rate in humans. That's what I said, post-puberty. And then the same number of mutations pre-puberty. So that intercept at puberty is the same between owl monkeys and humans. Okay, here's one question. You can all draw your own conclusions. Has the mutation rate actually evolved? So the average rate for the human data that I showed you, when they have kids at an average age of 30, is 1.2 times 10 to the negative 8th. If I change the puberty time and I change the average age after puberty that owl monkeys have kids, their rate is 0.82 times 10 to the negative 8th. And it's all explained just by sliding the line and changing the age, right? I don't have to propose that any proteins changed, that like proofreading proteins didn't change, cell division rates didn't have to change, nothing like that. So I think there's two ways to interpret these as, yes, the mutation rate has evolved, certainly it's changed, but that's solely due to these life history traits, not due to anything wrapped up in the mutational machinery directly itself. Okay, so is it all life history evolution? I think that would be boring. Does the mutational machinery ever evolve? So we're going to look at a second case where, of course, something more interesting happened. So let's look at Rhesus macaques. And if we think about how the mutational machinery might evolve, the number of mutations per unit time could change, right? And that could be because cell division is happening faster in one lineage than the other, or it could be because mutations, the repair enzymes are just worse at repairing them per, the same number of cell divisions, right? So if this is the human line, we can imagine in some species, the rate could get faster because cell division changes, or it gets worse at proofreading, or it could get slower again because cell division becomes slower or the proofreading enzymes become even better. We could also change the number of mutations that are arising pre-puberty. So I've treated this as like black box of development, but of course, again, we could change the number of cell divisions before development, or we could change something about mutational repair before development. And so again, we could shift it up or down compared to the human value, where other primates, other mammals might have more mutations or fewer mutations before that point. Just to make a point about how hard some of these models are to distinguish, I'm going to show you data from Mikkel Schirup's group. So the black dots here are, again, the Icelandic data we keep using, but now the other points are great apes. So I can't remember how they're assigned, but one is orangs, one is chimps, one is gorillas, right? But they're all very close in both reproductive age and puberty age to humans. And so I made up these lines, to be clear, but sure, if I slid a line over to the left, that seems like it fits pretty well among the other great apes. But if I change the slope a little bit, that could also seem like it fit really well. Or if I just like shifted it up and down, I could fit really well. So it's really hard to do all of these things in the great apes because they're so close for so many of these parameters to humans. So that's just one justification for why we're over in the other part of the primate and mammal tree, where puberty times are much lower and reproductive times are much lower as well. Okay, so we largely did the same kind of experiment in Rhesus macaques as we did in owl monkeys. The pedigrees have a slightly more complex flavor, but it's mostly the same types of inferences we can make. This project, and essentially everything I'm going to talk about from here on, was led by a postdoc currently in the lab, Richard Wang. And I should also say that, I'm telling you these stories in essentially the order in which we completed them. And so lots of our statistical methods and computational methods have gotten smarter over time, largely due to Richard. And so here's just a picture from another paper we have about how to use information in these pedigrees to help us. We're the underlying methods to what I'm telling you are changing from a little bit from study to study, but they're always getting better, I hope. Another interesting thing about this macaque data set is that here I've uncovered the legend of the Icelandic individuals. One of the reasons they originally did this study is that older human fathers have a higher chance of having a child with autism than younger human fathers. And one proposed reason is that they're leaving more mutations, right? So if de novo mutation from father is driving autism, you would expect this relationship. And it turns out that Rhesus macaques are a model organism for autism and kind of behavioral traits in general. So at the California National Primate Center, where we got these macaques, they've been studying the behavior of the offspring for many years. And they're studying autistic-like behaviors. It's not autism in these macaques. And so they could actually go and say, do older macaque fathers have children with behavioral problems that resemble autism? And quick answer is no. Okay, so in macaques, older fathers are not more likely to have autistic children. So the question is, okay, well, do they also leave more mutations, as in humans? And here's the answer. So yes, older macaque fathers still leave more mutations, but they don't have, they're not more likely to have autistic kids or autistic-like children. So I think that kind of answers to the negative, this connection between de novo mutation and autism in humans, unless all of the mechanisms of autism are completely different in macaques. But as you can see here, the male number of mutations, the mutations inherited by the male parent is going up with age, again, in macaques, and in females a little bit, but it's not significantly different from 0. So again, very, very similar to in humans. The mutation rate now is 0.58 times 10 to the -8th per base per generation at an average parental age of 7.5 years. And again, males produce 76% of all the mutations. As it happened, and this rarely happens in this field, a different group sequenced a bunch of Rhesus macaque pedigrees at about the same time. And this is their results. They're basically the same. In fact, their results are within 5% of our estimate. And if you've ever dealt with mutation rate data, you're like, that's unbelievable because I could do it on 2 different days, and if the humidity differs, like it changes by more than 5%. So like I'm pretty confident in our calls. But anyway, same results there. But now if we compare the macaque rate of accumulation to this human model, if we ask what's the function, it looks quite different from owl monkeys. So now it seems like macaques have a lower mutation rate at puberty than do humans or owl monkeys. Okay, so here again, the black points on the right are the Icelandic individuals. Each dot is one of those individuals from a trio. And the orange dots are the macaque individuals, the offspring of the macaque trios. So we can explain this in a couple of different ways. It doesn't, again, seem like the rate of mutation with time is changing. Right? We can't say that slopes definitely aren't different, but they're not significantly different. So again, it's this relatively constant rate of change. But there's 2 non-exclusive explanations for what's going on. So one is that the reproductive longevity model is still correct. Right? It's the same accumulation post-puberty, and there's this separate puberty effect, but there's been evolution of the number of mutations pre-puberty, right? It's been shifted down relative to humans. Or it matches the total longevity model because actually the macaque data are pretty close to being on that kind of extended human line there. And we can't tell the difference at this time between those 2 models. So something has evolved, right? It's either in macaques, something pre-puberty has evolved to become lower, or the total longevity is correct in macaques, and that means that our previous results from owl monkeys just so happened to fit perfectly, but actually there was a change pre-puberty in owl monkeys, so that became higher. Okay, so one of those 2 things happened. Okay, so many conclusions of the macaque situation. There's more mutations as macaques age. This rate of accumulation, it seems to be conserved across species, same slope in basically everyone. No association between older fathers and autism-related traits, even though they are accumulating more mutations. And the number of mutations pre-puberty has changed in someone. At least it's changed in macaques relative to humans. We're not exactly clear yet why. All right, questions about macaques? You can pause for that. Sarah.
I'm sorry, but so you represented the idea that the rates would change over time with those 3 different slope lines, but to me, that's showing just 3 different rates, not a change in the rate over time. A change in the rate over time would
Yeah,
be
I don't mean to say the change in the rate over time.
Okay.
I mean what you're saying.
Okay.
Yes.
Okay. Got it. All right. Just making sure.
There's no evidence for any sort of non-linear slope to any of these things, even in humans, where we actually have power to say.
I was going to say that's something I'm super interested in, because going back to your intro where we care about mutation rates, one of the major reasons why we care about them is because they might compound.
Yeah,
so
no,
we would expect that.
No evidence of that.
Thanks.
I mean, I think the only caveat to that is we're not truly looking at one individual through time, right?
Right.
These are like a bunch of different trios, but still.
But we do have it across many ages, so.
I have a bit of a naive question. So I understand why it is that you see more mutations accumulating in the father than the mother because eggs are formed very early in development. But I guess my question is then, what is driving the mutation accumulation in females at all? If they're formed, in theory, before a lot of mutations have accumulated.
So I think the main hypothesis is that, the DNA is sitting there and it is accumulating damage.
Okay.
And so it's accumulating pre-mutations, and there is still one more meiosis step.
I see.
And that's, you're basically going to turn all those pre-mutations into mutations at that one additional step.
Gotcha. Okay, thank you.
Okay, so before I get to cats, I'm going to take a little bit detour and just, again, stress why it's important to know these functions and how understanding mutation rates help us. And so I'm going to return to this hominoid slowdown question, right? So why is it that nucleotide substitution rates are lower in humans and chimpanzees than baboons and macaques and marmosets? Maybe I didn't say, but owl monkeys are New World monkeys. So owl monkeys would be over next to marmosets somewhere. They would have this fast rate. And so one of the most important things we know in evolutionary genetics is that the rate of substitution over time per million years equals the rate of nucleotide mutation. So as long as we can get these both on the same scale, the nucleotide mutation rate per year should equal the nucleotide substitution rate per year. So K should equal mu. And so there's been multiple explanations for why you see this pattern. The kind of original model, which is completely wrong, but people were studying like Sturtevant 1928 because he had done a few experiments, was that like every species had a fixed number of mutations per generation. And if that was true, then if you had a longer generation time, as humans and chimps do, you'd have many fewer substitutions, right? And therefore the substitution rate would be higher in short-lived species than in longer-lived species. So you can plug it into some math, which isn't very important, but the answer you get out is mutation rates per generation are the same between species 1 and species 2, but the substitution rate per unit time is much higher in species 1 than species 2. Okay, kind of relating this model, which clearly isn't true, to the model I've been talking about. If you reason through the same things for the total longevity model, at least if you imagine that rate of mutation accumulation through age goes through the origin, then the rate of nucleotide substitution per unit time is the same between species, but of course the mutation rate is slightly lower in a shorter lived species, right? Because it's just this mutation clock that's occurring in every organism. And then the reproductive longevity model is like a model in the middle, because there's this little bit of fixed effect, and then there's some association with age. Okay, so again, there's an asymmetry in mutation rates and a slight asymmetry in substitution rates. So we can plug all of these. Oh, actually, I forgot. Molly Zavorsky has a paper that says it much more elegantly than I said it just now. But we can use my words, which is if we stretch this line of accumulation during post-puberty down and it goes through the origin, the rate of substitution that's on the bottom is flat. It does not change with age. If we stretch that line post-puberty and it hits above the origin, it can't hit below. If it's above the origin, then there's going to be some generation time effect. There will be some slowdown, right? So total longevity on the left says there's no change just due to life history traits. Reproductive longevity says there is some change due to this fixed period, right? That the fixed period is basically the offset from 0, why it hits not at the origin. And we can plug some of the relevant parameters, male conception age, when males hit puberty, when females have kids. And the purple dot at the bottom is owl monkeys. That is estimated to be the fastest rate per year. And macaques are estimated to be, that's the red dot, in an intermediate rate. and humans are estimated to be the slowest at the top. So we get a qualitative association, but actually our estimates of the rate are way off. So there's something about some of these parameters that have changed over the history of the tree to result in the differences in substitution rate we see, right? And using these models, knowing which model controls the mutation rate will help us parameterize what the changes must have been. Was it a change in the male conception age? Was it a change in male puberty, etc.? This is just a kind of a summary and a much more realistic tree of substitution rates from Priya Morjani. And you can see at the bottom are the nucleotide mutation rates estimated from pedigrees. Mouse lemur is a big outlier. And at the top are the nucleotide substitution rates. There's a kind of correlation, but it's not nearly as close as we would expect if we could explain all variation in the substitution rate. Just as an example of a place where we can say something definitive, for a long time, my lab has studied the rate of gene copy number change over time, right? So now imagine we're not talking about nucleotide changes, but the genes being duplicated and lost over time, or just parts of the genome being duplicated or lost over time. And there's a rate of change of that as well. And what we have found is that the rate of change of those mutations is highest for the hominoids. And it's at an intermediate rate for the other primates, and then it's at a much lower rate for other mammals. Okay, so this is the opposite of the hominoid slowdown. There's a rate acceleration of this type of mutation in the grade 8s, especially in human and chimps. And again, fortunately, Thomas Marquez Bonnet and Evan Eichler's group basically did the same experiment and found the same thing using completely different methods. Again, they found this speed up in the rate of gain and loss in the hominoids. Okay, so two different mutational mechanisms are one slowing down in this group and one is speeding up in this group. So we looked at the copy number mutation rate in Rhesus macaque, the same pedigrees I showed you, and we re-examined it in humans. And what we found in both cases is that there is absolutely no association with the age of the parents, right? So neither in macaques nor in humans do older individuals leave more mutations. And this actually makes perfect sense. And the reason it makes sense is that most of these mutations do not occur during mitosis. They occur during meiosis because they're due to homologous recombination. And there is exactly one meiotic event per generation. And so there's no reason we would necessarily expect for these mutations to accumulate as individuals got older. So weirdly, you know, copy number variation fits this fixed mutation model of this like one event that's the same, you know, per species, per generation. But the pattern we see is completely the opposite, right? It's not a slowdown, it's a speed up. So something changed about the mutational mechanisms of copy number variation. And it made it like the actual mutation mechanisms changed in the hominoids. And I can talk about what those reasons are another time. But at least we can say something definitive like, yes, the mutational machinery did change. Okay, I need to speed up a little bit. So cats, haha, this preprint came out today, which is good timing. So we looked again at domestic cats. It's a very young age of puberty, lots of individuals, lots of offspring, well-known pedigrees. And the cats fit the reproductive longevity model. So here now, there's 3 lines because I represented the reproductive longevity model as the solid blue and the total longevity model as the dash. So you can see that cats don't kind of get slid down the human line, they get shifted over. So if we want to break the tie between owl monkeys and macaques, cats say the reproductive longevity model is the better model. It's macaques that something shifted pre-puberty. And again, this rate of accumulation, the number of mutations that accumulate with age, Sarah, seems to be about the same or not significantly different in cats than in the other species. But we want to do something excess beyond that. So yes, it fits reproductive longevity. And so the next thing we thought about was, well, it's not just mutations, like the number of mutations that are function valued, that are a function of age. Actually, the mutation spectrum is a function of age, right? So here are the same Icelandic data, but now broken down into the 6 different types of nucleotide substitution if you collapse the 2 different strands. And then CPGs, which are hypermutable in mammals, are separated, and indels are separated. But what you can see is like they don't all change the same with age, either among males or among females. So the mutation spectrum, like the total accumulation of mutations that you'd see, changes with age. And there's been lots of work kind of comparing mutation spectrum among species. So here it is for owl monkeys on the top, and then humans and chimps on the bottom. As you were taught as an undergrad or in high school, there are more transitions than transversions. In all species, the CPGs are extra mutable. There's the orange ones signified at the top, but they differ, these spectrum differ a little bit among species. And the issue now is that, well, the spectrum is also a function of the age at which we measured it in all of these different species, right? It would change depending on what age we sampled individuals at. And so just as with this kind of analogy of tree height, we're now kind of thinking, okay, if I look inside the stem of these trees and I try to characterize the cells that made up the tree, they would differ, right? So these are actual pictures of some tilia and the xylem at different stages. And that trait itself is also evolving inside the organism, right? But it's just changing with development. Okay, so if we imagine how does the spectrum change? So if we kind of benchmark it at 30, we see that some mutation classes would go up and some would go down. If we imagine we had younger individuals, and some would again go up or go down if we had older individuals than the average Icelandic individual. And so again, we can use these two kind of contrasting models of reproductive longevity and total longevity to say which one explains the mutation spectrum. Are we measuring, essentially a human 3 years post-puberty, or are we measuring a human at 3 years of age? And those two would predict very different spectra. And here are the results. So the cat data is the black. The orange bars are the reproductive longevity model, and the blue bars are the total longevity model. And I will just say overall, the reproductive longevity model fits better. So you can see in the C to T category, they're much closer than the total longevity model. That's true of C to Gs. C to A are both equally bad. But for some of the other categories, maybe not A to C, total longevity fits a little bit better than reproductive longevity. And so what I think this means is that different mutation types are being ruled by different models, right? Some of them really do have this like more or less constant mutational clock. And some of them are being reset at puberty and then taking off from there. Okay, so mini conclusions, because got one more section that I want to use those ideas for. More mutations as cat age, again, the same across species. Same or not different slope. Same number of mutations pre-puberty in cats as in either humans or owl monkeys, but different than macaques. And the mutation spectrum, not just the mutation number, suggests that different mutation types may follow different models, right? They all may have slightly different clocks. Okay, last bit. I want to use all of these ideas to say something interesting about humans, about like the actual biology of humans. So one thing that all of this suggests, all of these results suggest, is that I could predict the age of a human if I knew how many mutations they left, right? I could basically say like, okay, well, I know how many mutations you left or were left to you by your parents. So I think, this was the age of your parents. And I could do something similar with the mutation spectrum. If you tell me the mutation spectrum of the new mutations that you inherited from your parents, I could tell you how old they were, right? But that's really boring because we can just go out and ask them how old they were. So I don't want to do that with living humans. I want to do that with humans who lived a long time ago. And here's the idea. So I can't get a trio of not extinct humans, but humans who are not alive today. I can't get a trio, but I could say, here are all the mutations that arose in this time period. And if I knew all the mutations that arose in a time period, I could tell you the average age at which people were having kids. So it's not attached to any particular trio, it's attached to a population of humans, right? You give me all the mutations from this generation in time, this slice of time. I tell you the average age at which people were having kids. So I need two things to be able to try that inference. I need data allelic variance. So I basically need to know when did each of these mutations arise, and be able to say, here's the mutation spectrum for this slice of time. And then I need a model of the mutational spectrum. I mean, I kind of gave you a model already, but I'm going to make the model slightly better. So luckily, there's all of these very smart people who worked out how to give us data allelic variance. So there's one method from Albers and McVean. It's GEVA is the name of the method. I don't know how they pronounce it, but it basically, for every segregating SNP in humans, tells me how old it is in numbers of generations, right? So at every slice of time going back, I can say, here are all the mutations that arose 1,212 generations ago. I mean, there's a plus or minus with those estimates. And this is the mutation spectrum of all those mutations. So I can get a bunch of variants and figure out when they arose. And I can build my model of the mutation spectrum from the Icelandic data that we have today. So basically what we're going to do is say, we're going to attach a vector of frequencies, the proportion of each of these mutations, to parents of different ages. So technically we're building a Dirichlet multinomial model of the mutation spectrum. So for every combination of male and female parental ages, they emit some proportion of each of these different mutations. So I get a bunch of mutations. I know they're from one slice in time. I have a model that can predict how old were the males and how old were the females that produced this spectrum. And so it looks a little bit like this problem, where I try to minimize the distance between my model and the data, except it's a 6-dimensional space and I have a parameter separately for males and females. But this is basically what we're doing. Okay, so I'm going to plot the data on here. Average allele age. We're going to go back 10,000 generations. That's the bulk of the data. The data gets pretty sparse after 10,000 generations ago. And then we've got the generation interval, which is essentially what's the average age at which males or females were having kids in that slice of time. So for these 10,000 generations, we broke them up into bins of 100 generations. So we don't literally say 1,212 generations ago. We say, you know, between 1,200 and 1,300 generations ago. And that's what the data look like. So males are in blue and females are in red. And then the average is down the middle. So for every slice in time, every bin, there's a blue dot and a red dot. That tells you the best fit for the average age at which males had a kid and females had a kid. And you can say that it was, you know, average age of like 30 for males and 20 for females, 10,000 generations ago. It went down a little and now it's swooping back up in the last several hundred generations. Just to put some kind of times on there, because it's on a log scale because of where all the data points are. I should have said that each window has the same number of SNPs. That's why it's on a log scale, so that we can account for the fact that our SNPs are of different densities going back. So, you know, somewhere around 70,000 years ago, a little bit more recent, the average generation time started going down and then it started going back up about 5,000 years ago. Here's a blow up of just the last 1,000 generations. So again, we just took this interval and cut it into 100 bins. And you can see, you know, generally the same pattern. Males always have a slightly older generation interval, average age at reproduction than females. The average goes down and then swoops back up in the recent past. We can also track that difference in males and females over time, and it goes down and then comes back up. And that's largely because it's mostly determined by males. So the male ages are bouncing around much more than the female ages. So the male-female differences have also changed over time. This is actually really consistent with anthropological studies of current societies. So they're not highlighted, but you can read kind of among nation states and among hunter-gatherer societies is how this anthropologist split them up. Males have longer generation times in like 97% of the cases. Here, they're also a little bit bigger for the hunter-gatherer societies, like the gap between males and females is larger than in current societies, right? So perfectly consistent with what we've been able to recreate from SNPs emitted by these individuals hundreds of thousands of years ago and thousands of generations ago. Okay, so just the conclusions of that part, the human generation time has changed over time, according to our data, but not too much. It's a relatively slight change. Consistently older generation times for males, which is also consistent with the biology of males and females. Males can have kids until they're, 60, 70, whatever. And so that's going to really upwardly bias the average generation time for them. And in general, the difference between the sexes is getting smaller as we get towards the more recent present. Okay, so just wrapping up all of it, how do mutation rates evolve? Well, sometimes it's just this shift in when do you hit puberty, right? Everything's the same except you become pubescent at 3 instead of 13, and everything else is the same. Sometimes it seems like actually maybe we're on that line, or some mutational types are on that line of this constant mutation clock. At some point, there's some changes pre-puberty in the mutation rate in some species. We didn't find any evidence for it, but certainly it's possible that we will eventually find species at which this rate of change changes over time. Just in terms of what we're doing next, we have projects underway to look at grizzly bears with Joanna Kelly, and they're interesting because they undergo hibernation, and we know the spermatogenesis stops during hibernation. So there's a really interesting question of what happens to male mutation rates. We're looking at possums, American possums. So it would be the first marsupial that we get an estimate from. And that ugly primate on the right is an II, which is a strepsirrhine, and we have very few estimates from the lemurs and strepsirrhines. We're also really interested in, well, what are the stages at which all of these changes are happening? And so we have some experiments planned essentially to look at all the early stages to pair estimates from the placenta with the grown individual to kind of isolate the first stages of development, to pair the germline with the grown individual to kind of get at the pre-germline specification rates, and really break down this complex trait into more manageable units so we can see what's going on at every stage. Okay, with that, lots of people to thank. In my lab, I mentioned Richard and Greg. They've led all of these projects and have been great. 2 undergrads, Artie and Jelena, have helped out on all of this. 2 new grad students who are beginning to help on these projects, Samer and Yadira, and a colleague who was in computer science here, Peja Radovoyets, helped us to get all of this funded originally. At the Baylor Human Genome Sequencing Center, Jeff Rogers, he's the primatologist. He knows how all of this runs, and they know how to sequence things. The macaque stuff was maintained and data was collected by John Capitanio, cats by Leslie Lyons and Bill Murphy, and owl monkeys by Lawrence Williams and Mary Galinsky. And thank you.
Thank you so much. Do we have any questions?
I have a question. I know you mentioned that you didn't want to talk about interspecific variation, but I only want to ask because there was this recent paper in bioRxiv that is essentially argues that the signals we see by calculating things such as the D statistic is actually an artifact of higher mutation rates in Africa instead of introgression, which, to some extent, I don't believe, because we have like haplotype evidence, like such as EPAS1, but I think it does add like an interesting counterpoint and like fun thought experiment. And especially when you showed me the different, that, mutation rates were slower in like hominoids, old world.
Yeah. Okay, I'm going to go back to the slide that I skipped over because I said it was too complicated. I can find it quickly. But okay, so first of all, like that's Bill Amos. Like he's been tooting that horn for like 10 years, whatever. Okay. There is absolutely no evidence that the mutation rates are higher in Africans than in anyone else. So the orange dots on here are different human studies. And if you just plot the average age of the individual studies versus the mutation rate they got, they fall on this human line. Right? Once you account for that, and other people have done this, there is no evidence for a difference there. There is evidence for a slight difference in the Amish, but it's like really, really small. That's absolutely not to say there's not genetic variation. Almost 10 years ago, we found genetic variation among fly lines in mutation rate. You know, there's got to be variation, but yeah, not in that comparison.
Okay, that makes sense. I just didn't know after this, I was starting to rethink that idea, but now that makes sense.
I got a question.
Yeah.
In your, yeah, it was a great talk on your estimate of the generation age going back in time in archaic humans, I guess. I'm just curious, well, two things, technical and I guess biological. One is that obviously that's from ancient DNA samples, right?
No. So these are all
This is all inference from standing variation in
all
humans.
inference from standing variation, right? So like they take
It's
the
like
current
PSM,
steps.
PSMC gone crazy, right?
Yes, exactly.
Okay.
You basically build a giant genealogy and you say, I know the mutation happened on this branch right here, and I can date that branch. Yeah, so I think like we thought about using ancient DNA and it becomes much more complicated because those still aren't de novo mutations, right? Those are standard mutations of the ancient samples, and there's DNA damage. And
Yeah, exactly. That was the DNA damage. And well, the question I had was, I guess would still be relevant to using extant and a sort of genealogical approach is I guess it comes down to sort of like David Pede's question. If there are different mutation rates between different sort of geographic populations of humans, and we have admixture in recent years, is that curve, that sigmoidal curve you showed for the changing generation time, a consequence of mixing over time rather than a consequence of actual change in some germline mutation rate?
Yes. So
if you're basing it on standing variation and different geographic populations of humans have different, pi and theta, whatever, then I'm just curious how that would influence your pedigree or your coalescent base estimator.
So I hope it shouldn't estimate it, right? Like even if the mutations come from Neanderthal, they're being estimated correctly in terms of age, right? Because we're not just using allele frequency. Like they're actually building a genealogy and saying, these are old mutations. Just because they entered humans, let's say from Neanderthals, or there was mixture between two human populations, doesn't make them suddenly seem younger. It just means that the shape of the genealogy at that locus is different.
Mm-hmm.
And so like the interesting thing is that even if there's selection, as long as selection is not eliminating mutations differently, right? Like there was a sweep, the genealogy is different, but we're correctly aging the mutations on it. So I would say that like the separation there between what's the genealogical effect and what's the estimate of the mutation age, those are kind of different problems. And hopefully mutation age isn't being affected. Maybe Amelia had her hand up and maybe she was going to ask something similar. I don't know.
No, I was just going to ask, so you used all the standing variations. If you go back to your genealogy, slide 91. So what the standing variation from what populations?
So this is the total 1,000 genomes project data. So this is brand new stuff that's not fully digested. We are currently trying to separate it by subpopulation, but at the moment, this is all populations together.
Okay, but you don't expect to see differences, right?
I have no a priori expectations of differences. The only possible kind of expectation is that I believe kind of demographically, like actual population demography, if you have a growing population, your average generation time is going to go down.
Mm-hmm.
And so I think that's what a lot of the slope downwards on this slide is about. And so, if we think about populations that, went out of Africa and then expanded, we might expect more of a dip in those than in African populations.
I see. Thank you.
But
That was a great
that's a
point.
guess. Yeah.
I have time for one more question.
I've got one if no one else does, but by all means, I'm the interloper. I'm really curious about your result about the CNVs, and I think it's so interesting that that may shed light on the different evolutionary operators that affect different types of mutation rates differently. And I'm curious, most of what you showed us today was using the base substitution mutation rates. And I'm wondering if you are at a point where you can do a lot of these analyses with the other types of mutation that occur to see if differences in the slopes or the
Yeah.
differences between males and females, et cetera, will reflect something to us about the differences of the mechanism or the evolutionary pressure on the mutation rate as a trait, depending on the type.
It's a good question. My answer is no, because it's really hard. It was much harder than I thought to get these CNV mutation rates. There's one problem is there's less than one per genome, per generation, right? And so you have to sequence many, many more trios to get even a manageable number. And they're much harder to call, right? It's not like all the allelic balance stuff that we, worked for nucleotides, like it was just not happening.
It's definitely harder.
And we've done this before, right? Like you and I have done this before. It's not easy.
Yeah.
And so,
but wouldn't that be so illuminating? I mean, that would.
Yes, I guess so.
I'm in the camp, do everything with base substitutions first because it's easiest to, but I'm definitely not in the camp that base substitutions are the most interesting or highest impact mutations that happen.
Yeah, I mean, I think, okay, so one thing is you'll notice I didn't give you a rate for these, right? I just said it doesn't go up with time. And like, it's because the estimates of the rate, the confidence intervals are so big, I don't even want to say a number.
Mm-hmm.
Like it's less than one per generation. Like that's the number.
Yeah. Yep. Okay. Your next life, next career.
Oh, it looks like there is a follow-up question to that. Catherine, if you want to, or do you want me to ask it for you?
I can see it. It's okay. Why did the CNV rate speed up in hominoids? That's a good question. And we've been trying to answer that. So the weird thing about the CNV rate is that it's because of non-allelic homologous recombination, right? Like those are being caused because things that are not alleles are lining up. And it tends to correlate with TE, bursts of TE activity, right? So TEs are the substrate on which non-allelic homologous recombination are occurring. And when you get a burst of TEs, you tend to get a burst of changes in copy number of the rest of the nuclear loci. And so it seems to us like what's going on in the hominoids is that bursts of TEs have led to temporary bursts in the rate of basically CNV formation. Hopefully that answered your question. But it's complicated and it's hard to measure.
Thank you.
All right. Well, thank you so much, Dr. Hahn, for joining us and sharing your cool research with us today. And everyone else, thank you for joining us. And I hope you all have a good evening.